Hello, everybody. We'll just wait a couple of moments for everyone to join us. Thank you very much for being here with us this afternoon. Just hang on just for a second as everyone's joining us, joining the call. Okay, so hello everybody. My name is Dr. Laura Ashram. I am the Chief Executive at the British Neuroscience Association, and it is my pleasure to welcome you all to this special webinar, which is hosted in partnership with our supporters, Scientifica. Today I'm joined by Geoff Ingram and Josina van Lunteren, who are product specialists at Scientifica and will be presenting today on the topic of overcoming the challenges of patch clamping. So we'd love for you to get involved in this conversation today. Do say hello in the chat and let us know where you're dialing in from. We'll have time for some questions at the end, so please use the Q&A function to submit those questions at any time throughout the presentation, and we'll get to them at the end. I'd like to take this opportunity to remind you that the webinar is being recorded with the permission of our speakers, and it will be available to watch on demand on the Scientifica website. So I will now hand over to Geoff Ingram to introduce our topic today. Thank you.
Fantastic. Thank you very much, Laura. Josina's just going to share the slides and we'll get started. Fantastic. Well, it's a pleasure to contribute to this webinar series, and we plan for this to be a very practical exercise in overcoming the challenges of patch clamping. Throughout the talk, there are going to be links and helpful pages dropped in the chat as well. And also, you guys, please do drop any questions that occur to you straight into the Q&A. Even if you think we might answer them as we go, it'd be great to see which things are coming up and occurring to you as we go through. Our hope is that this will be a useful nugget or two in here for you to help you make your patching lives easier. I'm Geoff. I've been at Scientifica for 8 years, and throughout those 8 years, I've been working with customers like you to design and quote these full systems. It can be a full system that was starting from scratch for a new lab or expanding a lab, or as well visiting you in your labs to review your current systems, understanding what it can do now and where you want to take it to. And then together we decide and work on upgrades and improving your systems. Josina is a product specialist just like me and an ex-patcher who did a PhD using Scientifica kit too. So that's our background and why we reckon we're in a good position to give a practical overview to help you. But then why Scientifica? Well, Scientifica's been doing what we do since 1997. First, we started distributing the Burley manipulator, and then we designed our own patch-star manipulator, and that's grown to full EFIS rigs and even multi-photon systems. We followed our customers through that journey. Scientifica is headquartered between London and Brighton, right between the two in the south of the UK, and we manufacture everything just outside of London in Maidenhead. We focus on electrophysiology and imaging. That's our world, and our aim is to be the most trusted provider of these systems. So who is this webinar for? Whether you're new to your PhD and just getting started, or if you've been at this for years, maybe even decades, we hope there'll be something useful for you in here. If you can leave this webinar with an idea to try on your scope that might solve a problem you've been having, then absolutely that is job done as far as we're concerned. So Scientifica is a manufacturer of full electrophysiology rigs. So let's address these challenges from the hardware side of things. Let's imagine we're starting our patching journey and we're just sat in front of a rig that's just become ours. The question is what sort of microscope is this? Is it going to be an upright or invert, as we see? So the question is going to be based on whether you're working, say, in culture, monolayers of cells. If you're doing that, you can use an inverted system where the objective comes from beneath. You can access the cells easily that way, and it practically reveals a huge amount of space above your sample. Easier to get your pipettes in, and contrast techniques and things tend to lend themselves on these systems too. If you're working in slices which are relatively thick, you need to then access them from above using an upright microscope. So the practical difference there is that the working distance of those objectives is going to be down into the millimeters. So it's a little bit more challenging to really get your pipettes in, but then that's where kit like this comes in to try and make that as easy as it possibly is. When you're looking at these systems, you can then subdivide them a little bit further. You have a patching staging solution that you can build around another microscope, typically Olympus, Nikon, Zeiss, Leica sort of microscope, or you can have dedicated systems from Scientifica who basically we provide the full system, do it fully motorized as well. So on the very right-hand side, there might be something that's a little bit new to people, and that is our new solution for patching in cultures, essentially our inverted system. So now let's dive into the sort of key bits that as you're staring at this new system you've got, what's actually making it up. So predominantly this is a system that's going to aid you to visualize your cells and bring your patch pipettes down to the cells. So first things first, we're looking at the microscope itself. The questions are, what sort of brand is it? How old might it be? And ideally, who used it before you, if someone did? Be a fantastic source of information, quirks about the system and its general capabilities. Then next is going to be sort of the staging and the motorization. You've got lots of different options for different staging in this world, whether they're high up with everything mounted on the same platform, things low down, asking questions like, are my manipulators and my sample, are they linked with the staging or do they move independently? That'll affect how you can move around your sample as you get moving. And then fundamentally, does the microscope move or does my stage move? The next thing is your objectives and the objective changes. Now, in the world of upright microscopes for patching, you'll likely have two objectives that easily flip between or slide between. One's a relatively low magnification objective with a fairly high working distance that helps you get situated and begin to bring your pipettes in. And then one is a higher magnification, typically then lower working distance objective 2. Questions are the objectives that you have appropriate for the type of experiments you want to do? Is the trade-off with magnification and a shortening working distance worth it for you? Then next is going to be your condenser and your contrast. You'll need contrast on a system like this to be able to see your cells, and there are several different types of contrast that are typically used, all of which are adjustable and work really well with a range of samples. There's no best option, no correct option as such. Cross-referencing the different components using Scientifica's knowledge base is a really good way to tell if you're not sure. And mainly, particularly if you're using differential interference contrast, which takes a lot of parts, the question is, are all of the bits there? And are they all in the right positions to actually generate the image that you're attempting to get? The next section you can sort of look at is eyepieces. And there's a big usability point do you have these eyepieces where you can look down the scope as you're working on, say, a fairly manual system? Maybe you have micrometers to move your stage, you're manually adjusting in Z. It can be ergonomically quite nice to be there and looking down. But if you're in a world where your microscope's particularly motorized, you can be sat over your computer a lot more. What we see is there's a bit of a sort of camp you fall into. It's which one you started with tends to maybe prevail as a preference. I've never seen swapping from a scope with eyepieces to a camera really trip anyone up too badly. But the thing to remember, if you're on a scope that doesn't have eyepieces, you can probably add them if you'd like. And while we're talking about cameras as well, is huge range of cameras on these sort of systems. The question is the camera up to the right spec for you? Do you potentially want to do things like calcium or even voltage imaging now or in the future? Can the camera you have do that for you? And is it going to work into the future with software updates and things like that? The next thing, of course, is the manipulators. What manipulators do you actually have around? Are they in good condition? Have they been mounted in a way that works for you? Do you have the room you need to interact with them? And can you swing them out in all of their glorious ways for adjusting and changing pipettes? And as well, do they do things like move at a comfortable speed for you? A lot of manipulators have the ability to go into software and adjust the speeds. So you can really hone them to be ideal for you rather than just living the way they are. Then next is epifluorescence, a reflected light path on your system. Now, you may or may not have this on your system, and you may or may not need it. If you do have it and you do need it, understanding how your light source works and if you have the right tubes to work for the wavelengths you need in your turret is going to be key and easily upgradable. Then kind of getting into things like the inline heater and perfusion. There's a lot of questions about how has it been set up? Is it working nicely for you? Does it have the features you need? Or is it even too complicated? Potentially there's been drug applications been done in the past on a system that isn't necessary for you now. Do you need to use a complicated diffusion system, or can you use a more simplified one? You streamline the system and have it less complicated. Then, as a final point, the data acquisition. There are lots of options here, and understanding the capabilities of your setup will be key for sure. Understanding whether it's you're in the camp of Axon or Hecker, or even using open source softwares and things as well. So with a bit of that sort of large funnel down from the full systems, I'll hand over to Josina, who will take you into even more detail.
Thank you so much, Geoff, for the introduction on the system in general. And I will continue with a little bit more introduction, and that will be more on patch clamp in general. So I'll start out with the different patch clamp techniques. Because Geoff already touched on this, right, when he talked about inverted and upright microscopes. So inverted microscopes are often used with cultured cells. When you're talking about patch clamping in cultured cells, this is a very practical technique. You can use many different sort of cells that you can then electrically classify or characterize. You can also adapt these cells in multiple ways, and you can easily apply all kinds of compounds. You can even measure single channels and what is the effect of the compounds on these, and this is very often done in cultured cells. So this is a specific type of data. And then if we move on to the slices, so this is another type of sample that is often patched under an upright microscope, like Geoff said. Slices are usually fresh brain slices. So you take a brain and you keep the slices alive. And in this way, you can patch a cell or multiple cells that are still in the natural environment that can communicate to other cells in the slice still. So you get a lot of information about the cell. And you get information about how the cell is connected to other cells or how it interacts with other cells. And then there's also in vivo patching. So one can do the patch clamp technique in vivo, where you can patch a single cell inside an animal model, for example. And then, of course, you get the real information of the cell in the brain with all the interactions with the brain still intact as they are. And this can even be done sometimes in awake animals, or you can do some stimulation even if the animal is anesthetized during the experiment. So this is, again, a different type of data. And then, of course, we talked a lot about the equipment that we use already, and we will talk about it way more. And this is all to get your patch pipette to your cell. And then when you are at your cell, there are also different ways you can record. So if we look at the first picture, this is cell attach, where you just bring the patch pipette to the cell and make a nice giga-ohm seal on the cell membrane. And then you can listen to the activity of the cell. Basically, if you see spontaneous activity, you can see it like this. And the advantage of this is that you won't disturb the cell at all. But to influence the cell, to control the membrane potential of the cell, you need to break the membrane. And you need to break into the cell. And then you have full control over the membrane potential, and you can test how it spikes at different potentials and everything. So this is you get very valuable data out of it, but you will diffuse the liquid from your pipette into your cell, so you disturb the cell more. So there's also a way where you can do perforated patch with a perforating agent in the pipette, where you can kind of get best of both worlds. So you don't disturb the cell so much, but you do have some control. And there are different configurations where you take a patch of membrane and you only measure what's going on in this patch of membrane. And the advantage here is that the inside of the membrane is now facing the bath, so you can you have an easy time applying compounds to the inside of the membrane, so you can study certain channels in that way. So yeah, these are all kinds of different techniques that give different data. And then I want to talk a little bit about how you get this data, because as Geoff mentioned at the beginning, I did my PhD, and I patched in my PhD on a Scientifica scope also. So I just wanted to take you through like how the patch comes to be, how you do the patch lamp experiment. And I decided to take this beautiful flow chart that was made by Nasrin Chowdhury for our website to help me guide you through it. So to start, you need to fill the micropipette and put it around the electrode on your rig. And this is already a little bit of a challenging process because everything is very small. And on the Scientifica rig, it's very practical. I always used these features of the patch stars that I had, where you can turn the patch star towards yourself. And I also used this, yeah, you can see properly. I use this feature where you can pull back the head stage, so you can easily like change the pipette. And there are some more features we will talk about later. So you kind of like turn and take the pipette and wiggle the pipette around the electrode. So it makes the point is to make the contact between the pipette and the intracellular medium that you have in your pipette. And this is what actually makes the contact, because that's a salt solution. So this is what makes the electrical contact between the cell and your data acquisition. And that is the start of that contact, that measurement. So this is important to get it right. And then to then approach the cell, you need to find the pipette under your scope. I always did this in the highest magnification, and I think some people do it in the lower magnification, then switch to the higher, but I always did it in the highest. And I had this student that was telling me like, oh, you must have very good eyesight, because I was kind of staring at this pipette like if it was going underneath the magnification, because if you know your rig, you know when you can see when your pipette is in the right place. But I don't think you really see it. It's more the knowing of the rig. So this can be a bit tricky if you are on a new rig, or if you switch between rigs, you always have to make this way a bit. And then if you have that, you're gonna move down with the pipette and the objective to a cell you picked. And you always move down with the objective first, because if you would move down with the pipette first and you would go too far, you would destroy the cell you picked carefully, maybe because you would hit the tissue accidentally, or you would destroy the pipette, or you would destroy both, and the recording would not happen. So you carefully go down objective first and then pipette. And I always did it by hand with the wheels, so with the motorized microscope and the manipulator, but with the wheels. But you can always also let software do it. And so on the Scientifica scopes, there's a follow function. And you can use that to just make the pipette and objective go down at the same time. And then when you approach your cell, when you see your cell and your pipette, you can approach the cell and you see the dimple, and you take the pressure off of your pipette, and you hopefully get a good seal on your cell. And then you use the least advanced part of your $100,000 rig that you have. You use or many people use a syringe that they have taken apart, and then they just use their mouth to break into the cell, to apply suction to the cell to break the membrane. And I also did this always by the suction by mouth and everything. It worked very well for me because you get a feeling for this, and this is like, if you know how this works, this is the best, of course. But then the pandemic, of course, hit. So it took a pandemic for me. But then I decided to change my ways and go little bit more advanced. So I took a complete syringe, and I learned how to apply pressure with the syringe, and with the three-way stopcock, you can control this very well. So that's the things you need. And it also helps to have a little measurement, a little pressure meter in your tubing, so you can see the pressure on your pipette. That is very practical in many situations. I will probably mention it later. And then if you have broken into the cell, of course, you can get a recording. And then to get data, you do this many, many times over. So Nasrin already put all these little things that can disturb you or can go wrong. And today we will look into a little bit more like challenges that can exist on your rig or that are a little bit more persistent, that you cannot step over, so you have to solve them. So today we hope to give you some tips and some key points maybe to look out for on things that can be a challenge on your rig when you're patching. So we will look at drift. And then we will also look a little bit at electrical noise, what to do about that. Then we will have a quick look at the temperature controller. Then we have some tips on like starting to construct your rig or placing your rig in the right place. And then we have some tips on finding the right cells and getting a nice image, and also finding cells with fluorescence. So the first thing I want to talk about is then drift. So our Scientifica manipulators are of course made not to drift, but still, if you find that your pipette is drifting, there are some things that you can try to just make it go away very easily. And when I say drift, I mean that if you are looking through your microscope, because the drift will be very small movement. So if you're looking through your microscope, then you see your pipette move in relation to your microscope. So they don't stay in the same place. And what is, of course, these are precision instruments. So what it's meant to do is you put it in a certain place. And if you stop moving the wheels, the pipette and the microscope will stop moving. And this is how you keep a stable patch, right? So what to do if you see your pipette move? Then you can check if it is the manipulator that is moving. And then for this, you should tighten the bolts on the manipulator, and also have a look at your manipulator if it looks like it's mounted straight, if it's not skewed in any way or anything. And that's important for the manipulator. And also on the manipulator, you have the locking stops. So I can point them out. So in this sliding carriage, for example, there are locking stops, and there is this little thumb screw that one should, because I always did this, right? I took that out when I was changing the pipette. So for every new patch, you need to tighten that. And if you forget it, then you will not have a good time. So this is important to remember. And also there's the rotary states here and the base of the patch star, on the patch star, that have locking stops. And these are magnetic stops, and they allow you to turn away the manipulator from your experiment. But when you're doing the experiment, you should make sure that the magnets that make the stop, like that contact on the stopping point, that they're in the right place. So then the manipulator will be stable when you're doing the experiment. So you can change these places where they stop with these little grub screws on the side. So you can completely customize them so they're perfect for you. So this is very advisable to do, so you don't risk doing an experiment in the position where the manipulator is not in the most stable state. And then on the head stage, you can also check the pipette holder. The pipette holder holds the pipette, and it's just a little plastic device that holds the pipette, but it's sealed by two O-rings, by two little sealing things on both sides. So you can check if these are tight. Maybe they need to be replaced. Maybe the whole thing needs to be cleaned so it holds your pipette firmly. It also has to be tightened properly every time you change the pipette. And then another thing that can shift is, of course, the microscope or the recording chamber. So you wouldn't see the difference under the microscope what is shifting, if it's the manipulator or the microscope. So you should also go through the bolts on the microscope and the recording chamber to see if that's all stably mounted and if everything is correct there. And then a thing that I've seen cause drift also multiple times that is a bit more difficult to control is the temperature changes. So it can just happen that a manipulator drifts in the hottest week of summers of the summer, then there is, of course, not so much you can do, but you can always check if there is air conditioning that you can use, so you can keep the room at a more constant temperature. Or maybe there is air conditioning in the room, but it's blasting straight onto your rig, so your rig gets way too cold when it's on, and then it turns itself off again because it has a thermostat in another place in the room. And then your rig heats up again. And the same goes for heating. There can be heating close to your rig, which causes temperature fluctuation. So this is important to note these things and to try to fix them if possible in the place where you're patching. And then last but not least is the cable management. So here we have this beautiful picture where you see many cables and you think, oh no, what a mess. But to me, the cable management is good for the manipulators because the cables attached to the manipulators are loosely hanging off and they have some slack. So the manipulator has the freedom to move into the sample and you have the freedom to do the movement you need for the patching, and the cables will never pull on the manipulator. So the Scientifica rigs, they also come with a little box full of screws where you have this little black plastic holder that's very practical to screw down onto an air table, and you can clamp the cables to the table with that. So that they don't like slowly start falling off the table and pulling on your manipulator. That happens also in cases. So yes, then the next thing I wanted to discuss is electrical noise. So here you can see our poster that we have for electrical noise. And this lists just a list of steps that you can do to find electrical noise and to eliminate it then. Because electrical noise can be a bit daunting. It's a bit magical. It can come from many places. It can appear on certain days and disappear other days. So my first advice is to look at this poster or look at this list that I have here, and then just try these steps, and also to ask for help then, in addition, maybe from if you have someone who is more senior to you or has some more experience, you can always ask for help if you are in that situation, of course. But the first things to do are to use an oscilloscope, or even just there's in the PClamp software, there's a pulse test that just generates pulses, so you can see what's going on in your signal, and you can then with this you can recognize wavy patterns that are there, because noise often comes from peripheral equipment, and this will give 50 hertz noise in Europe and 60 hertz, of course, in the US. And you can just recognize this period, and you can see that there is some noise from electrical equipment on your oscilloscope. Or you can maybe see that it's not likely to be from electrical equipment because it looks very different. So you can already see a little bit what is going on. And then to eliminate this noise, it helps to just systematically switch off and remove and unplug all the peripheral equipment. And then, especially if the rig has been in use a bit, or maybe there were different users before you, it may help to redo the grounding wiring, because you can create loops in the grounding, especially if you keep adding new stuff on, of course. And then if you put back the peripherals, you can see maybe if the noise comes back, and you can determine what is causing the noise, and you can try to isolate this, place it farther away from your rig, and that should help. And you can do multiple rounds of this, because if you have a big source of noise, it could be masking a smaller source of noise. And you will only find it when you solve the larger source of noise first. Then I will skip a little bit ahead, because when you're doing this with all the peripherals and you have a lot of trouble with this, you can use a Faraday cage, and then you can place power supplies and like these power bricks that do the conversion for certain equipment. You can place them always outside the Faraday cage, and the Faraday cage then shields your rig and your experimental area from the electric noise that comes from these components that have to generate electricity to run your rig, right? So this can be very practical, especially in persistent cases where you don't have the possibility to get away from the noise. And then you can also check the perfusion system, because the perfusion system is often a long line with salt solution, so it will pick up electric noise. And you can, for example, check if there are cables right next to it. That's usually not very good if there are power cables right next to it. Data cables don't matter, but power cables. And you can even ground the perfusion system. I've seen my colleague do it with a needle, for example, through the tubing. Or you can ground the heater that also sometimes solves the noise. And Last but not least, you have to check the pipette holder again, and also what you're doing in your experiment. So the pipette holder, the seals on the pipette holder, if they're not tight, like you could like have a small movement in the pipette that you haven't seen, or something could leak, or the pressure could change. So it's important to check these things. And also, if the pressure changes especially, but maybe there's something else that's happening, if you're approaching your cell and trying to make a seal, and for whatever reason your seal isn't very good, then you will also see a lot of background noise, of course, in your recording, which can just come from your seal not being very good in the beginning. So it's good to pay attention to that, if that could be a cause. Yes. So then I have a short note about the temperature control. So we at Scientifica, we sell an inline temperature controller, and there are only a few things that you should pay attention to. The most important thing is because this heats up the bath solution while it's coming into the bath. So it heats up the line where the bath solution comes through. And it's important to place the heater then, so this little part, as close to the bath as possible, so you don't get a lot of temperature change, of the liquid on the way to the bath, right? And there's also this probe that will measure then the temperature in the bath. So this is how we keep exact control of the temperature in the bath. And with this heater, you can set some settings in the software that tell the software how to adjust the temperature if it detects a temperature change in the probe. And there is a standard setting that works very well to keep the temperature very beautiful and constant, but you have free reign over this setting, so you can change them, for example, to make sure that your bath heats up faster if that's something you would need. So then maybe the temperature will be a bit less constant for you, but if you needed to heat up fast, you can do that. Or maybe you were forced to put the heater farther away from the bath, and maybe you can adjust for that in the settings. So those are things to look for if you're struggling with keeping your temperature constant. And then I said, we will talk a bit about like how to set up your rig and where to set up your rig. So if you're starting to think about patch clamping, it can of course be quite daunting to know where to start and everything, or to build a rig from scratch. So there are several things you can do. First off, we can help you because at Scientifica we sell complete rigs, so we are a one-stop shop for all the EFIS stuff. So we can talk you through the whole rig and tell you like what is possible and where you can start and everything. But when we're doing that, we are always asking for your current needs and your future plans. So we will not just tell you everything about the rig. We will always ask you like, what do you want to do with the rig? And then we can specify how the rig is set up, depending on what you need to do with it. And it's also always good to think a little bit about the future, so you can maybe add things on that you will not need right now, but you need in the future, or you can check if the rig is upgradable, so you can already prepare it to be completely upgradable in the future. So on that note, even if you don't need a completely new rig, but if you need to move a rig, or if you want to revive an old rig that is there, then we can also help you to get the old rig up and running again, and add some things on so it can do what you need it to do right now. Like, and then it's important that this rig that you build, it will be intuitive to use. So the Scientifica rigs are usually praised for being intuitive to use. And you can also adapt them to your needs, like I showed in the software with the temperature settings. And Geoff mentioned also with the manipulators, you can adapt everything, all the settings, so that they work for you. And this can be very good for new students in the lab that want to learn how to patch, and they need to get to something, of course, that works, and that is also easy to understand for them, because it's difficult enough to learn how to patch. And we can also come to your lab and do full installation and training on your rig, so you will actually end up with a working rig in your lab. But before we put the rig in a lab, it is also important to check, or maybe you're moving the rig, so you want to put an existing rig somewhere. It's important to check certain things in the environment. So I thought to list them here. So the available workspace, that's the first thing, of course. You can even adapt the rig to fit in the available workspace. You can say like, oh, I have great future plans and I want to do a lot of extra stuff on the rig, so I buy a very big table. But if you don't have the space, you can also say, I want a very small footprint rig and I buy the smallest anti-vibration table that I can fit there, and then I make sure the rig fits, and we can help you check if it's all gonna fit. But always next to the rig comes usually a rack with equipment, and also a computer, and some person, an experimenter who sits there. So you need to take this into account when you're looking at available workspace. Not only the microscope has to stand there. And then there's some other environmental factors, like I already mentioned the heating. So if you are in this position that you're gonna set up a rig, then before you pick a room, you can check if the heating is adjustable freely, and if there are any like very acute places where it's very hot or very warm, you're in a certain stream or airflow that will mess with your equipment, but even maybe with the cells that you're trying to pet. So it's not good for the sample usually to get temperature fluctuations. And then another thing that we didn't mention yet are windows. So this gets more important, I think, when you want to do fluorescence imaging. Then you need to have some control over the light. So if you have a room with many windows, then you need at least the possibility to hang some curtains so that you can control how much light comes into the room during your experiment. That's quite important. Or you can avoid windows, of course. And then another thing to avoid is walkways, and especially busy walkways, because you have the rig on an anti-vibration table, and this will usually cancel out any vibrations from the occasional colleague that walks by. But if you're in a very busy walkway, you get problems with vibrations and airflow again, and maybe even distractions to the experimenter. So it's much better to be in a more quiet place. And then I want to hand over to Geoff again, because the Airtable, of course, filters out many vibrations, but Geoff has some tips what to do if you notice that your anti-vibration table is not filtering out everything anymore.
Absolutely, exactly. So the main thing that people go straight to often when an anti-vibration table isn't working, isn't acting as that complete disconnect from the world around you, is to start questioning, okay, is the pressure correct? In my experience over the years, for the most part, the pressure can be really very forgiving. If the table's floating nicely and it's not, vibrating almost itself, which would be very easy to notice, the pressure is typically fine. What you're mostly going to see with one of these tables, if they stop working, is a mechanical short somewhere in the setup. Now, if you have a Faraday cage, these are typically either mounted on top, which might act a bit like a sail, which can start things. So addressing that, maybe moving it off the tabletop if you can. But sometimes you have a perimeter enclosure that goes around the whole size of the table, and then the Faraday cage sits on top of that. Even a really small connection, a cable resting, or things like that can cause vibrations to be transferred. So going around gently, these are relatively heavy tables and there's a finger trap hazard, but go carefully around and see if you have any mechanical shorts, first of all. That's likely going to snap and solve the problems. The next thing would be kind of looking underneath the table at how the table's leveled, and if everything is floating as it should be, with about an even gap on all of the legs. They typically have 3 legs that you can adjust, and there's little arms that come out from each of the pistons. Pretty self-explanatory how you adjust them, and just have a gentle, sort of move with them and sort of see how they react to the tabletops. Have a look at the pistons themselves. If you sort of see they're kind of pulled over at an angle, that could also be causing mechanical short inside the mechanism. And at that point, you'd sort of either be looking at sort of knocking the top back into place, or ideally bringing lifting gear in to lift the table up, straighten the pistons as the table's off, and then gently resting the table back on them. If you have any questions about that sort of thing, or really anything along these lines, feel free to reach out to Scientifica. We're always happy to have a chat and go through by video call, just a quick phone call.
Perfect. Thank you. Yeah, and then I will do the last point, which is the question, is your rig set up ergonomically? So we have to, of course, take into account that there is going to be an experimenter who is doing the experiment, and they need to be able to reach everything. So they have the computer that they need to interact with a lot, and also the sample and the microscope that they would need to interact with a lot, with some then remote controls, usually. So they need to reach all this without bumping into everything all the time, because that will destroy the experiment. But they also need to sit for quite long hours at the rig. So it's also good if they can sit in a proper position while they're doing all their work. So yeah, these are the things that I would say are important to keep in mind. And of course, I said multiple times, you can call us if you need any advice. So there is a QR code for you there for a lab consultation. Then the last 2 points I wanted to talk about have to do with getting good image. So on a PET lamp microscope, there is usually a transmitted light path. And so always a transmitted light path. So it's the light that goes through the sample that shows you the sample. So the light comes from one side of the sample and then goes through a condenser that focuses it on the sample. And then it passes through the sample. The sample changes the light. And the objective then forms an image that, with this changed light from the sample, this is the way you see what is going on in your sample and what is a good cell and what is a bad cell. So there is a simple trick to optimize this transmitted light path. So Geoff already mentioned that there are like different contrast methods to visualize the sample, and these contrast methods are applied in this light path. But for all these contrast methods, there is a condenser, and if you set the condenser right, you will get the best image out of this transmitted light. So to set the condenser right, you first would focus on your sample. It's called coloring, by the way. So first you would focus on your sample with the objective, so you just have your sample in focus. And then you're gonna set up the condenser correctly. So to do that, you close the field aperture, which is the little black knob on the Scientifica slice scope. And then you just have then an iris closes and you just have a small little point of light that is coming through, and you can see that. So then, because you can see the edge of the iris, you can focus the condenser by focusing the edge of the iris. So if you see a sharp little edge, you will have focused the condenser in the correct way. And after that, you're gonna open the field aperture. Oh, no, sorry. After that, first you can center this little dot that you have in the middle of your field of view. And then you can open it. And when you open it, you can check if it's properly centered. That's why I went too far ahead. So when you make it bigger, you will see if it's centered in your field of view, of course. And then to go back to imaging your sample, you open it until it's outside the field of view. So you have your whole field of view filled with light. And then you have this light that is coming to your sample through the condenser. You have it focused and centered on the place where you're doing the imaging. We also have guides on this, so you can go through it step by step if you haven't done it before. And then there's another type of imaging that many people use, and it's fluorescence imaging. So this is very handy to find certain cells, because you want to patch a certain cell type, and you can mark this with fluorescent markers nowadays in many models. And then you will get way more data from this certain cell type that you're after, right? Because you can easily find this cell. So to set this up, you would have to use the right fluorescent light source that fits your fluorescent marker. So the fluorescent marker has a fluorophore associated with it. And this fluorophore is excited by a certain wavelength of light and emits another wavelength. So you need to match the wavelength of the light for your fluorescent light source with the fluorophore that you're using. If you don't have a light source yet, if you're looking for a light source, then you can, for example, call us and say, I want to use these fluorophores, and then we can help you find the light source. Also, the other way around works. If you find out what kind of light source and filter sets you have, you may be able to get the right marker into your sample and image it correctly like that. And I just wanted to highlight that if you're starting this, it can be a bit tricky because you have two filter sets for this, and one will show you the contrast image, so that's the gray image that you see here in the background. And the other one, the other filter set will show you a fluorescent image. And here you see a nice overlay, but when you're at the microscope, you have to switch between these filters. So first thing is there's usually a little screw or adjustable thingy, so you can make the filter set switch more smoothly. So there's usually a wheel. That's advantageous because then you will get less vibrations in your microscope while you're approaching your cell, and it will be easier. And then to check in the beginning if everything is going well, you can take pictures. That's what I did here. And then post hoc, you can overlay these pictures, and you can see if your pipette was at the correct cell that is actually fluorescent. That's a very good check in the beginning. And if you want to do more advanced things, of course, there are ways to fill your cell and to do post hoc stainings where you can put your sample under a fluorescent microscope. And then you can see maybe even other genetic properties of the cell, or you can see if you got the right cell, if you PET the cell that you expected, and even see what the cell looks like morphologically. So there are a lot of opportunities with fluorescent imaging. So I will leave you with that idea, and I will hand over to Geoff, who will tell a little bit more about our role in this.
Absolutely. So as Josina mentioned, we developed these systems from scratch to work extremely well. So a lot of these challenges that come up in your pursuit of patching are addressed in Scientifica equipment. Grounding's easy. The manipulators are rated for less than 1 micron of drift over 2 hours. So bearing things like that in mind when you're looking at your equipment, asking a lot from it is something you can do. If you're looking at a piece of scientific equipment that isn't working 100% to spec, that means we can put it right. There's something environmentally that can be solved through this, through drift checking, through servicing of equipment. These precision instruments, and they last for many, many years. Perhaps at some point they need a little bit of service. So it's all there to be assisted with. And like I mentioned earlier, you can go from adding patch stars, staging around an existing microscope you have, upgrading, or speaking to us and getting entirely full systems.
Yes, and then we go.
And as I say, it's the support and the ongoing relationship that's really important to us as well. So we have this resource center, which is a really vibrant resource for you guys to use quickly. And as well, reach out to us. We're here to help, as well as a dedicated support team to help you forwards as well. So here's a chance to answer some of the questions that have come through, and we've got some brilliant questions. So thank you for sending those through. If anyone's got any more, drop them in and we'll run through. We've got sort of 10 minutes or so to keep chatting. Before we crack on with the questions, there's a poll that's about to come up asking if you'd like to be contacted after the webinar. Feel free to click that if it's interested in talking about buying any piece of kit. But also, if you'd just like to chat a bit more about some of the things we talked about just offline, feel free to drop a yes there, and we'll just give you a call and sort of discuss what you're really interested in and how we can help. So please do click and answer that. And I will jump into the questions going from, so we've got 3 so far, so I'll start from the most recent and work back. So the question is, so we need to switch between the contrast and fluorescent filters in order to patch cells. Not necessarily, it depends how you're identifying the cells you wish to patch. Potentially, if you want to use if you want to use a marker to identify your cells. You could identify them and then swap to your contrasted image over fluorescence and patch that way. But potentially you can actually get a dual filter set that lets you visualize the fluorophore and visualize your infrared transmitted light. That's something you can add to the system. Anything to add, Josina?
I think that filter set also works for optogenetics, right? So you can do, because the optogenetic, if you want to do optogenetic stimulation, you can use the same light path as you use for fluorescence. So on an upright microscope, because then I can explain more clearly, this light would come from above because it's the reflected light path. And if you have then a filter cube that lets through the light from above, but also lets the contrast through to your camera, you can do the optogenetics while you're monitoring your cell basically with your camera. So that's also a handy one. Yeah, and you have combined filter sets also for fluorescence. Yeah. But I think if you want multiple colors, you are turning on many microscopes, you have to turn. So yeah.
Yes,
something to keep in mind that sometimes this is necessary. Yeah,
exactly. Sometimes you will be switching filters. One thing we do quite regularly at the moment is make use of quite powerful LED fluorescence illumination systems. You can do that one way with many different LEDs with specific wavelengths, but also you can do broad light sources and add filters to the LED itself. That lets you put a multi-band filter set in your microscope and then actually change the fluorophores you're looking at by changing the wavelength of light sending to the filter set. We design those all the time and can do some really quite powerful experiments because of that. If that's interesting, we can talk some more.
And that can be automated also.
Yes, exactly. TTL it out from your protocols and everything. It's really cool. So Baptiste asked the question, let's say the heater is heating at 60 degrees and that the length between the heater and the bath. Won't the cell slice be damaged by throwing a 60-degree solution onto it? Yes, I noticed that in the shot we showed, the maximum heater setting is set to 60 degrees.
That's too high.
Well, it very likely is. But what we're looking at here is in the Scientifica heater, we run a feedback loop. So we've got temperature sensors both in the heater itself and in the bath.
Oh, the heater.
Yeah. So what we have is actually that max heater setting can really be a way of you making sure you will never throw extremely high temperatures on yourselves, because you're quite right, that would be disastrous.
Yeah.
What you have is when we talked about setting your heater as close to your sample as possible, it's preferable. Mitigate noise and things, use a Scientific heater that's designed to make that extremely easy. But by placing it very close, what you do is you have less loss between the heater itself and the bath. So you don't have to set the heater temperature, or the heater doesn't have to run and heat the temperature up to say 60 degrees because it's losing so much before you get to your target of say 33. It does a feedback loop and cleverly adjusts. What you might see in these settings that we used here, where the heater is very hot, you might see a lot of degassing and bubbles coming out. So that can be a challenge that you sometimes see with inline heaters. Changing the settings and making sure the maximum heater can't go too high can be a way of actually mitigating that as well as protecting your cells as well. Anything else you've seen, or Baptiste, comment again if there's anything further you'd like clarification on that.
Yes, please. I'm happy that you are reading out the questions because I haven't found them yet in my screen share.
That's okay. No worries. No worries.
I have nothing to add to this. This was perfect.
Cool. So a question that came through very early on in the chat. Baptiste, thank you. Glad we answered the question. From someone at just 60 minutes in, is there an opportunity to give any hint of what kind of control we need to use? Now, in my mind, I hear control. How we work the movement of the scope. But I think that question is likely talking about controls in the actual experimental side of things. So very much over to you, Josina.
But what was the question? How to incorporate controls?
Yeah, essentially, that's my interpretation.
But then we need to know more about the experiment, I guess.
Mm.
Do you know
And
which?
what did you tend to use?
Yeah, so I when I used controls, I usually had two different groups of mice. So there was one that had a genetic modification and one that was wild type, for example. So those were my controls. Right, so that would be a good control. But if you're working more, oh yeah, also if you're working on wild type mice, maybe you would want to see the effect of some drug or compound and you would wash it on. And then you can do kind of a within cell control, I guess. So you can run a protocol before you wash on the drug. And then you can run the protocol while you wash on the drug. And then you see a change. And then you can, with certain drugs, you can even try to wash them out again. So you can see if if it changes back to what you saw before. So this is also a control. Do we have more controls in PetSclamp? Please give me inspiration, chat, because now I
No,
ran out of controls for PetSclamp experiments. There can also
absolutely.
be, because I spoke about two conditions, right? Like there's a wild type and a mutant, but you could have 3 or 5 conditions. You just patch more cells before you say you see a difference. Yeah.
Nice.
Um,
Okay, so there's another question that's just come through is, hi, any experience or tips for patching inserts? I have 300 micron thick inserts in the incubator and an inverted microscope to deal with. Is it possible to patch in these conditions? Um, I'm I'm a bit stumped on that one, to be honest. I hate to give a negative answer. Um, because yeah, 300 microns thick would be a typical slice thickness,
yes, if you
which
have.
we would typically put onto an, an upright microscope.
Yeah.
Um, I guess as trying to be practical and be helpful, maybe my practical brain clicks in here. We sort of say to look, okay, let's say it is difficult to achieve the the patching you want to do with your particular sample with the equipment you've got. Um, let's not despair, but let's assess what you've got in terms of the equipment, because there can be a lot of things that you can pull from an inverted microscope, depending on what type it is, to an upright to be able to help you achieve the experiments, sourcing other equipment that's already in the building as well. And that's the sort of thing we can totally get involved with. So I would say maybe if you haven't clicked yes on the chat yet, hit info@scientifica.uk.com. Ask
Yes.
that question again, and we can find you, because I think that was from an anonymous person, so we better
Oh,
to track you down. But we'd love to help out and see if we can be helpful in the future.
yeah.
So okay, we've got a bunch. So let's zoom through. We
Yeah.
might actually run out of time. Thank you
No.
for asking so many questions. Really appreciate it. Let's go. Hi, I've grounded everything in our rig: light sources, pumps, tubes, with inserting needles, etc. However, I still cannot eliminate some of the electrical noise. Any thoughts on what could be the reason for this?
Now I have to give my thoughts, right? So we have
Please.
grounded everything, but did you also check, like, if all power supplies and everything, if is there a Faraday cage, and are there any things that could like introduce noise again that you can still move outside of this Faraday cage? Or yeah, I
Hmm.
don't know. Is there a bunch of power cables that runs together or something? Or do you have a little coil of power cables still lying next to your and something like that? So like I would advise to move things more away from your rig, like all the power supplies and cables. That would be my first advice. But maybe you have done that already too.
Perhaps the idea of something like a video call with
Yeah.
someone from Scientific here. We can have a look, another set of eyes on the system, and see if there's anything that we can catch. I wonder if you use any hum silencers or hum bugs
Yeah, maybe.
to maybe in the system. If you have, say, a digidata from electric devices for the Axon setup. They sometimes come with inbuilt hum silencers, which are designed to get rid of mains buzz. You can add those externally via a humbug addition, perhaps, as well. But yeah, reach out to us. We'll have a chat and give you.
Yeah, and wait, there's one thing I should add. That was also on the slide of the, but if they say they grounded everything, but they cannot pinpoint, like, have they tried like turning all kinds of things off and on and everything? So if you can pinpoint one thing that is causing the noise, you will have a smaller problem, right? Because you know what parts you should solve. So that could, yeah, maybe help. If you can say it is the perfusion, or it is this one, or this one thing that I added last week, or I don't know, something. Yeah.
Brilliant. And maybe we can squeeze in just one more unless we need to cut off. Let me see if we've got. They're all anonymous, I'm afraid. So please reach out if we don't answer your question. We'd love to chat. But do you have any recommendations to make a seal in regards to pressure?
Yes. Make a seal in regards to. Okay, but your pressure depends on your sample. And we usually apply the pressure with a syringe. And I knew on my syringe and in my tubing how much I should fill the syringe and how much pressure I should put, right? So the tip was to put a little meter with like in line with the tubing system you have. So you can at least read the pressure so you know when you're applying less or more pressure, and then you can test it out, right? Also, did you know you can take off the pipette holder, so the little plastic thing that's on your headset, you can take it off and seal it completely with a pipette, and you can dump it in a beaker with water, and you can do like you would do with a bicycle tire and see where bubbles come out to see if there's any leak and you are losing pressure over time, right? So these are the things to check. Like try to measure it and check if there are any leaks. These are, and then also look what your tissue does. Like I pet in slice a lot, but if you're blowing everything away, you have too much pressure. But this depends. Like if your mouse is 2 weeks older, you may need more pressure. So yeah, look at what looks good and what feels good. Yeah. This is it. Do we have time for one more question, or no? We should probably stop.
Very nice.
Soon.
We should probably wrap up. But it's really interesting to see the questions that came through. Thank you to everyone who joined us and you sent questions in. And actually, people said some really nice comments in here. So thank you for that too. There's one final question about patching HT22 cells. The key issue there being very thin.
They're very thin.
Yeah, any tips for patching thin cells like HT22?
I'm not the best with patching. Like I never patched very thin cells, so maybe I'm not the one to ask. Actually, then I would lower the pressure so you don't blow them away. Start there, maybe. Oh, you could even adjust the size of the pipette. You are free to adjust the size of your pipette to the size of your cell. So if they're very thin.
Approach angle potentially as well.
Approach angle. Your
making more acute.
Also gets more important.
If you're able to, based on the setup and the working distance you have, with, an objective perhaps. But yeah, I feel like we've not given a full answer. So please reach out again, and we can
feel free to
a little bit more about
get more answers.
Like.
And maybe actually here the tactics is also, maybe I know someone who has more experience, a customer that we can connect you with. This would be a good tactic also in this case.
So given the time is running out, we've got a few questions from Emery, Connie, and Federica. Thank you for saying that's you. I'll make a note of these comments and we'll reach out to you to answer these questions later on after the webinar. But thank you for sending them through. So fantastic. Thank you, everybody. And yeah, I suppose.
Thank you so much. We're signing off.
Thank you so much.